Wonderful. 9:30. We're all here. Welcome to Neuroccino. The way it works is that you present a paper that got you excited last week, and you tell us why you were excited about the paper and what they actually did, and then we briefly discuss it. Now, I know that today we have 3 papers if we manage to get through them all. So if you want to share your links in the chat so everyone can have a look at the paper. And we have Valentina, who's presenting a Neanderthal paper. We have Victor, who is presenting 10 Tesla imaging. And then if we have the time, Leah has a clinical paper for us. Is that right? Okay. And Michelle has an entire book. I'm not sure we have time for a book today, Michelle. I would push it to next week if that's all right with you. Right, okay, in the interest of time, let's crack on. Valentina, I promised you last week you're going to go first because I cut you short. So off you go.
You all see my screen?
Mm-hmm.
Cool. So this paper has brought to my attention by Stephanie, and it's about a study that quantifies the difference in endocrinal development between Homo sapiens and Neanderthals. So Homo sapiens is our specimen. According to previous studies, the endocranial before birth and post-birth development between Homo sapiens and Neanderthals have been species specific. So according to previous studies, they developed in different ways. So this study collected data on basically skulls of neonate, infant, and adult, both Homo sapiens and Neanderthals, and reconstructed endocrinal space according to a very large number of three-dimensional landmarks. If I'm not wrong, it was 922 three-dimensional anatomical landmarks that have been mapped onto the endocrinal space according to geometric morphometrics. And then they run a PCA test on these endocrinal landmarks to see how they are clustered together. And the PCA result showed that we have 2 very big clusters that represent the Homo sapiens and the Neanderthals development, endocrinal development, and then many clusters that represent the various phases of the development. Then the authors run multiple regression to look at the difference between the various endocrinal development clusters between the Homo sapiens and the Neanderthals. And the results show that at birth, so the neonatal endocrinal dimensions at birth is actually different between sapiens and Neanderthals. But then there are no differences in the overall development and the endocrinal development of the 2 species. And here they represent how Neanderthals and sapiens and endocrinal dimensions develop from neonate to adult. And as we can see in this figure, so in the early stage, we have positive expansions of the temporal and frontopolar cortex and the cerebellum. While and this is true for both Neanderthals and Sapiens, while for the late development, we have less expansions, basically, of the endocrinal dimensions, both in the adult Neanderthals and Sapiens. So this is really important because if before we thought that Sapiens and Neanderthals developed in a really different way, now we know that even though at birth we are different, we were different, in the successive endocrinal expansions, we actually follow a common path. This could open up various like speculations. One of those is that Neanderthal wasn't really like a specimen, an isolated specimen, but maybe with their similar endocrinal development as the Sapiens. They probably had similar cognitive functions, so similar cognitive capacities as the Sapiens, and they maybe have inbreeded, so they maybe entered like Sapiens societies and inbreeded with Sapiens more easily because of the shared cognitive functions that the endocrinal development suggests. And that's it.
Thank you, Valentina. Michelle, there you go.
This is absolutely great. I was thinking, so if Neanderthal and Homo sapiens are different at birth, but follow the same trajectory of development, this is going against the Evo-Devo hypothesis, which will be at least, you know, maybe not in utero, but at least at birth, which would be like we start the same, but we evolve differently according to species or along evolution. Yeah, there will be more like along evolutions. And yeah, this is going against that. So what do you think?
Yes, this is a totally new information, and I think actually I think that the authors expected Neanderthals and Sapiens being different at birth, as far as the endocrinal development is concerned.
But then they follow the same trajectory of development, so that's literally the opposite finding. It's funny.
Yeah.
Very good. Very good.
Yeah. I wonder
comment.
how many craniums do they have of Neanderthal? Because it's not like you have dozens of cranium for each different ages, I guess. So
exactly. So they grouped neonate, infant, and adult according to like broad range of age, because they had 70 here, 79 modern humans, so sapiens, and 15 Neanderthals.
Yeah,
and they were different in age, and they grouped the three groups according to ranges.
Mm-hmm. Okay, so it's quite a small sample.
Yeah, it is. I mean,
how do you have any information about the validity, the estimated validity of the finding? Do they know how much they are sure about the difference in the young ones? Because the most obvious difference to me, at least to my eye, is the neonatal different brains, and I don't know how many specimens they have to compare them exactly, because if they are
yeah, they provide a list of how many for each group, you mean?
Okay, yeah, for the young ones.
Yeah. So
modern humans, Neanderthal, 1, 2 weeks.
yeah, I mean,
okay.
So yeah, it's 1-2 weeks. I think it's
one per yeah.
Just one against 23 of the other group.
Okay. Yeah, so maybe they found something very important, or maybe the fact that it's very different at a very young age might be just chance for this specimen was the specimen they had just happened to be a bit different from what we expect. And
so you say it might not be representative of
yeah,
the Neanderthal populations.
Yeah.
Yeah, that's a really good point. They should have mentioned in the limits. But still, I think that such a study is really it's really new. Like nobody recreated the endocranial 3 dimensions of the brain of Neanderthals, and
yeah,
I mean it's also like means availability.
That's true. No, the study is great. Actually, it's I find it super interesting, and the way they tackle the problem is, yeah, very very interesting and cool. But yeah, that's just the
yeah. Still, there's a population representation.
they can't do anything about it. I know. That's yeah.
Leah, you had a question as well. Go ahead.
Yes, thank you, Valentina, for presenting. And maybe you said at the beginning, but I miss how they obtain the 3D representation of the skulls. Did you do like a CT through optical imaging?
Exactly. They start from the CT image. So yeah, they start from the CT image, and then from there they set like 3-dimensional markers, and started to map according to geometrical correspondence. Like they start with one sample of like neonatal one, and then from there they move to the second group and see the how the landmarks shift within the CT scan.
Okay, thank you. Yes, I missed it.
Great. Are there any other questions? Yeah, Victor.
It's more about it's more a comment than a question. It's about the fact that these there was another article about Neanderthal and brain very recently, but that were using organoids of brains to study the difference between Neanderthal and human. So I was wondering if you had seen it.
No.
Okay, I just sent it to the chat. They basically where's the chat? Here. Okay, so I just looked quickly right now because I just had the headline in memory. But basically, what they did is to compare the genome of Neanderthal and humans, and they created, I think, some kind of artificial genome of Neanderthal and created for the brain, and created organoids, like small little brains of Neanderthal, to compare what happened at the development stage of these organoids between Neanderthal and humans. And the results show that it's very different, at least visually. It's like the human ones are smooth, the human organoids are smooth, and the Neanderthal ones are very bumpy, for the lack of better term. So It was just, I just wanted to plug this in because I thought it was relevant.
Yeah, thank you. I'm reading right now. Yeah, that's cool.
It's like a live review of a paper.
Yeah. Thank you, Victor.
You're welcome.
Very good call.
Looks like Valentina has a paper for next week already.
Follow up.
I think the idea is good. So as so if you remember when I presented my the paper, this is different approaches. So the one from the endocranial and the one from genetics is very different ways to get to the Neanderthal brain. So very interesting to see the both approaches.
You could even 3D print one for us.
Yeah, and yeah, I had a. It's more, yeah, like Victor, it's more a comment, but I'm always wondering on the sample of Neanderthal, the secular effect on these samples, because I don't know. So Neanderthal has a spread from 30,000 years ago to 100. And so I don't know the effect of comparing a brain of 30,000 years to another brain so from the same species, but from very different time frame. So I don't know if they took that into account, because when you take a sample of Homo sapiens, you know they are from the same generation or very close, but for Neanderthal, maybe that spread over more than 1,000 years. So I don't know the effect of that.
No, it's a very good point. As far as I recall, they don't address this point, but I might be wrong. So I will give another reading and then I will get back to you, because it's a really good point. I mean, yeah, they compare like very different samples in terms of ages.
Yeah, and also the location. So, and in general, in terms of life. So, here we live in kind of very uniform society and a world. But for Neanderthal, maybe living in one place and live to another place, it's completely different. So we can imagine the development of a brain and the environmental effect can be completely different. So I don't know if they can take that into account, because it's, I think, difficult to go back in time. But
yeah, I will give it another reading, because I don't remember they addressing this point.
yeah, I think it's difficult anyway. So
Lovely, thank you so much. Right, let's move on to the next one. Victor, you ready?
All right. See the article?
Lovely.
All right, perfect. So this article is about, it's a news feature from Nature, but from 2018. So, but it's still kind of relevant today. It's about the strongest scanners that we have available, strongest MRI scanners. Because and in general, they are talking about the ultra high field MRI machines that are now, I wouldn't say common, but that are getting there. Like the 7 Tesla are really getting this. They really are starting to be available to a lot of research team, and these 7 Tesla scans are even more available thanks to, for example, the Human Connectome project that have made these scans open to the community. So the article is presenting the difference, the kind of the global scheme of these scanners from what are the scanners available to, what some of the problems and what they can do. To just to give you the overview, it's they say that currently we have 3 different scanners that are above 10 Teslas, which is absolutely huge. There are 2 in the US. So one at the University of Minnesota, and one at the NIH in Bethesda. And the third one is the one at Neurospin that you probably have heard of. And so the one at Neurospin, I think it's the only one for the, oh no, maybe not, I'm not sure. I know that the one at Bethesda is only for the brain. The one at Neurospin is a whole body one. And I'm not sure about the one at Minnesota's university. But they are all above 10 Tesla and are starting to ramp up the magnetic field. I mean, we already have images from these machines, at least for the one at the University of Minnesota and the one at I think a Neurospin already did a few tests. I don't know if they have already images. I have to check. But anyway, these fields, these machines have been created to try to go deeper in terms of resolution in the on the scans, because the principle of the MRI machine is that you create a very strong magnetic field in one direction that will align the protons inside of the water molecules, the spin of the proton in one direction, and then you send a radio frequency pulse in a perpendicular direction, usually, or it can be either, it can be in different angles, but let's say perpendicular for the example. And that means that the spin of the proton that you have aligned will start to wobble slightly and go back to their original alignment. And doing so, they will emit radio frequency that you can detect. And this is, depending on where they are and what tissue they are in, this is what you will detect. Actually, it's the spin, the wobble, and the going back to the alignment thing that you will detect, and that will give you information about that. Thing is, the quantity, the fraction of protons that have been aligned in the B0 field, so the main magnetic field, so this fraction of protons depend on the strength of the field. For example, I don't know the exact number, but if you, let's say you have 10% of the protons that are aligned under 3 Tesla, if you ramp up to 7 Tesla, you might have twice that. And if you ramp up to 10 Tesla, you can increase even higher, at a higher number. And the more protons you have, obviously, you will have more signal and less noise. So a better signal to noise ratio, which will increase the quality, the potential quality of your image. So this is great in theory because it will allow you to increase drastically either the resolution, the spatial resolution, you can go for smaller voxels, or to increase the temporal resolution, because you don't need to scan for as long to have an equivalent quality of signal. So in both cases, in theory, it sounds great. And this is exactly what they are trying to do. So for now, what I can say is that the resolution that they managed to reach with the 10 Tesla from I don't remember the University of Minnesota is that they imaged, I think at least they say in the article that they image the knee of a participant, that to check if they could see the fine details in the knee, and to check for to see if they could reach a resolution that could detect small changes that would help for the diagnosis and the treatment of arthritis, I think. And yeah, they really can get to a resolution where they can see something that is usually out of reach. But so yeah, so this is all great for in theory and get you to a much better resolution. But there are quite a few problems that comes with these kind of machines. The first one is that it is because you get to very finer resolution, and at least spatial resolution, it's much more susceptible to motion, the participant's motion, because you won't have like in particular for the brain, if you I mean, the brain is a very squishy organ, and if you start to move a little bit, or if you the example they give is pretty great. If they say that if you like clench your toes, because it is linked with nerves and the neural spine to the brain, the whole way in, it will slightly deform your brain, and this is usually not a trouble at all for the resolution we are at, but it will do a significant change in 10 Tesla or above MRI scanner. So yeah, so there is always the problem of the motions due to also the blood flow changing with the heart beating, and so yeah, a lot of small changes, small motions that would usually be just not really a problem are starting to, they have to take them into account. And so this is a whole new research field to not to just realign the brain when the patient moves, but to realign the brain when the brain moves itself, like it deforms under different stress that are like inside of the cranium, not from just moving your cranium. So this is one problem, but there is also the problem that are linked directly with the machine, like the fact that these at higher field, these for fMRI at least, these very high field are not as homogeneous as the one we have at lower field strength. And so this can be a problem for like EPI and stuff like that. I'm not super, I'm not a physicist, so I don't exactly know all the trouble with the sequence of the fMRI sequence and the problem that that the homogeneity will lead to. I know that inhomogeneity will lead to distortion of the image usually, and it seems that it's even worse in this case. So it's another challenge, but I'm sure that this kind of thing can be worked out. There is also the other problem of overheating the tissue, because we are still we are using very powerful magnets and strong radio frequencies that can and are slightly absorbed by the human tissues, which cause heating. And one of the challenges is to absolutely avoid creating heat spots inside of the brain, for example, or any organs, but that's really a danger. So they say that there are already strategies in place, and that it should be all right. But for now, they are not. There is no. The machines that they have are only for research purpose, and we are far from having machines at this high, these strengths for clinical for the clinical world. So Yeah, a lot of new things that seems to be interesting. Oh, there is also the fact that these kind of machines are extremely, well, they are very big, very expensive, but also kind of fragile. For example, the one that I talked about that was a Bethesda, the NIH one. It's like they broke it. It was received in 2013, I think. Here the magnet, and they broke it in the next few months that they had it, because they switch it on and off too fast. It caused an overheating somewhere, and they had to send it back to the factory to be remade. And at the time of the article, they were only receiving the new ones just a few months back. So, or a bit, or later this year, I don't remember. So yeah, these kind of machines are extremely expensive, hard to make, and require very technical staff to maintain it, and you have to really be careful with this kind of thing. So I think it's great, and they also want to keep advancing to higher and higher fields. It's very interesting, but I don't know if this will be the future of the clinical approach of MRI. It sure will be used for research, but I can't see it ever reach the markets of the clinical approaches for wider population.
Lovely, thank you very much. Super interesting. General question for everyone, but I guess our physicists in particular. Is the solution really to go higher and higher? Or would it be better to move more lateral and improve sequences and diversify the sequences? What do you think?
I mean, for me, I think it's something that is already being done. It's not like there is only effort on one direction. I'm sure that there are a lot of teams that are working specifically only on sequences. The teams, yeah, it's not something that, like, yeah, these kind of things, they do make the headline because they look exciting, but I'm pretty sure that a lot of labs are just working on better and better sequences. We just don't hear them as much because it's not as new, but yeah, pretty sure there are efforts in this direction too.
But just like you said, you don't see this being implemented in the clinic in the near future or slightly further future. It seems that by ramping up our Tesla strength, we're also increasing the problems. And the question is, does the gain in resolution, for example, justify the push for higher field strength and more complications? Or should we just come up with a different idea?
I think it's still necessary in some points. For example, one of the things that makes me excited about this is when you get to the resolution of the cortex layer, and then you can study the different layers of the cortex and have a relationship between the different areas, functional areas that you have. That is not just area A and area B are functionally connected, they are activated at the same time, but you can see it's layer 1 and layer 5 of these two areas. So there is a flow of information from one to the other. And that is something that I don't think we can do with 3 Tesla, and we can start to do it at 7 Teslas. But to get a real nice view of this kind of very fine structure of the brain, I think we still need to get to a higher signal to noise ratio, and so a higher field. It won't be for the medics, for the clinical setting, but it will be for the research, and I think it's still important to get there. Not necessarily to go higher and higher, to go 20 Tesla or something like that, but I mean, there are 20 Tesla for animals, but for humans, I don't see it. But I think it's still something that is necessary for the research aspect of the field, but won't reach the clinical aspect of it.
Leah also had a question.
No, yeah, linking to that, then I have also a question, but like linking to this, like in the clinics, for the clinics, I don't know about 10 Tesla, but 7 Tesla actually is used, for example, in really like specific cases. For example, as Victor was saying, you can really have information in the cortex layer, and that is useful, for example, in some pediatric cases where they have epilepsy, pharmacoresistant, and they really don't know how or where to make the surgery and how to stop this. And so in these cases that you have like a limit in the 3 Tesla or 1.5 Tesla, and like there are some clinical applications also in I know like about the 7 Tesla in Italy, in Pisa, where I went, and they use that for pediatric cases and also for Parkinson's disease to really see where there is the neurodegeneration, and so to then tailor the pharmacological trials. So there is a clinical application. I don't know about 10 Tesla or 11, but for 7 Tesla, there are many of these clinical applications. So related to research or really specific cases, because as you were said, motion is a problem. You really have to do like a training. Like I was impressed that there is a 0 Tesla where you just you are trained to go inside the scanner, because this required like half an hour, because it should be really, really slow also for your blood, for everything. And so it's a really like specific and like high level clinic research collaboration, but there are these clinical applications. And then I wanted to ask you also, it's related to this clinical application, if maybe there are some sequence that can, I don't know if it was the point of the article, if there are some sequence that can gain more about the eye field, for example, for angio imaging, as there was in the cover, like for susceptibly weighted images. You can also use like different coils, for example, for sodium, because the radio frequency change if you also change the so the resonance frequency change when you change the field. And so like you can have like also information about these things that we can have with 3 Tesla. There are a lot of problems, but there are new perspectives that can be used for the clinics actually.
Mm-hmm. Yeah, I've seen that they talked about the sodium imaging, yes, in the 11 Tesla, saying that they were managing to image thing. Oh no, it was for the 7 Tesla, because there is a whole part of the article about 7 Tesla, so I mixed a little bit of it. But yeah, I think yeah, they did some services, a lot of sodium imaging with it, and pretty interesting too. But just to add about the clinical applications of 7 Tesla, they are, I think it's pretty interesting. The example they give is not about the functional application, the functional MRI at 7 Tesla. It's about just the structural MRI at 7 Tesla, which is so much more resolved that you can see much finer details, in particular with respect to the veins and the blood vessel in general. And they said that it was used, as you said, it's for very specific case, but for this one, it was used for the implantation of Parkinson.
Deep brain stimulation.
Hmm?
Deep brain stimulation.
Yeah, for the deep brain stimulation, when they implant the electrode, they have to go through a whole part of the brain where there's a lot of vessels and stuff like that, and they have to be really careful. So first, it gives them the ability to know exactly where are the vessels and where they are trying to implant it. And also it gives them a better, it seems that sometimes they don't really exactly know where to implant the electrode. So they have to go fishing, for lack of a better word, like they try to plant it at somewhere and they record and they, oh, it's not here, and they remove it and they plant it somewhere else. And every time you do that, there is a risk that you puncture a blood vessel, and that would be really bad. So and they say that with the kind of image that they have there, with the precision they have, they exactly know where to put the electrode with these scans. So they just can say, okay, we go there, we here, it's good, done, no more fishing. And yeah, so that's I think it's a great application. I don't know if it will be used that often, but it's just a good example of what kind of thing this kind of precision offers.
I'm glad you didn't become a neurosurgeon, Victor.
I would poke the brain.
Oh my
God. Good career choice here. We also had a comment over on YouTube on exactly that point, which was that it's also important to focus on developing hardware optimized for sequences, rather than putting, like for DTI sequences, for example, rather than putting all our resources in developing pretty T1 and T2 images. So again, a bit more rounded, just more than pretty pictures.
That's true.
But you're absolutely right. Like the anatomical definition is just quite stunning. And for people who like anatomy, that's quite exciting.
Yes, I was also thinking that another application for 7 Tesla, I think that somewhere like in New York, in the USA, they do a lot of studies about multiple sclerosis, where usually lesions are around small vessels, small veins, and sometimes you can saw that in the clinics, and also it could be like a way to make a more precise diagnosis, because sometimes you can have also an overlap, or you don't know why this lesion is there. And so it can also help to understand more pathologies in that way, yeah.
Right, any more comments on this one? No? Let's move on to the next paper. In that case, Leah, take it away.
Okay, can you see, I think, the paper? And so this is an article published in Brain last week by Vidal and colleagues, and is focused on survivors, stroke patients, 135, who had selectively damaged the left hemisphere, and regions that involve the Broca areas and the surrounding regions. Maybe I will show you the group variability map of lesions. So were excluded patients with lesion on the inferior parietal lobe, and also mostly also the temporal lobe, because the studies was focused on the understanding if the Broca area, so Brodmann area 44 and 45, if they were responsible of long-term speech impairment, and or if it was a combination of all the other regions involved, and also if the white matter underline this area just above the insula and towards the frontal lobe. They focused on the anterior segment of the arcuate fasciculus. If also all these regions will affect the long-term speech impairment. So they select patients that they evaluated patient after 3 months after the stroke event, And as they were saying in the beginning, also in the Paul Broca seminal cases, we were like, he was like focusing on the Broca area, but these cases also have the lesions also in the underlying white matter and surrounding cortical areas. And so they want to investigate this aspect. And they use multiple regression. They use atlas-based. So multiple regressions was for the statistical approach, and also some Bayesian statistics. So a combination of these two, and also an atlas-based approach to evaluate the they call the lesion loading of these different regions. So the percentage of damage of Brodmann area 44, 45, pars triangularis and opercularis, and the ventral premotor cortex, so designating the precentral gyrus in those regions who are like more ventral and close to the arcuate fasciculus course, and also the superior central insula and the putamen. These were the cortical region used. And for the arcuate, for the white matter tracts, they considered the arcuate fasciculus, the anterior part. They just focused on that for their hypothesis, so lesions closer to the Broca areas. And but they say that, of course, the arcuate, the anterior part and the long segment are quite packed in that area, so they don't exclude an implication of both the tracts and the frontal axon tract. So they use these regions, atlas-based, and evaluated the loading according to the lesion, the stroke lesion of the patients, and run multiple regressions, considering, so here you have the picturing of these regions, so cortical and subcortical and white matter tracts. And they evaluated so the possible influence of these loading of these regions. This is the lesion overlap map to compute the lesion load. And they used different models to see which combination of these regions mostly predict the long-term speech impairment. They used for the clinical test the CAT, the acronym was the comprehensive aphasia test, where they used a stock picture description task, where it was shown a picture and the patients were asked to describe in 1 minute what it was happening in the picture shown, and some tips that in the study you can for helping the patient was what about death, for example. And they also scores above 60 were considered impaired, and also they controlled for the CAT semantic memory task to see if these deficits were more related to the semantic impairment, let's say. And so evaluating, they use different models to, as I was saying, use different combinations of the implication of the damage of these specific regions, and evaluating just the, let's say, Broca area. They saw that it was a significant, the Brodmann area 44, even if the correlation was not, the R square was not so high. Then considering, so they drop out the Brodmann area 45 5, just keeping the 44 and all the other regions. They say that considering all these variants in the model, it was significant just the arcuate fasciculus anterior segment, and with, let's say, a good correlation, a good R-square, 0.5. And then they also, so they focused on the arcuate fasciculus, and they considered again, the Brodmann area 44, and look again to the significance of each variant. And again, it was most significant, the Brodmann area, the arcuate fasciculus region. Then also they consider different possible combinations, and also like splitting the Brodmann area 44 in dorsal and ventral. But again, in these, according to these statistics, the arcuate fasciculus anterior segment was the one that was most significantly contributed to the prediction of the deficit. So then they also split the groups to see if there was, according to the lesions in these two specific areas, so arcuate fasciculus and Brodmann area 44. They create 3 groups if the lesion load was mostly on the Brodmann area 44, mostly in the arcuate fasciculus, often both. And they saw the significant differences. Where was that? Between, I mean, it was this one. It was. So they did. They found a significant difference in the between the arcuate fasciculus group and the Brodmann area 44. Just, but not when they were like together. For example, between the arcuate fasciculus and when there was an involvement of both the arcuate fasciculus and Brodmann area 44. So it seems that if the white matter damage play a significant role in this long-term impairment, and also they, using the Bayesian statistics, they wanted to evaluate the evidence in favor of the null hypothesis, that was that the damage of the Brodmann area 44 does not explain the variance in speech production, and it was a bias factor of 0.1, let's say. So it seems that it cannot really explain this long-term deficit. And on the contrary, the arcuate fasciculus damage seems to have a positive evidence, so a bias factor of 4 of this linking to long-standing effect of the in speech production. So to be sure, in the conclusion, they say that they don't want to say that the Brodmann area does have any impact in the for language impairment in stroke. Actually, of course, there is a link. A lot of studies show that. They also discuss other techniques like electrostimulations in some during awake surgery in glioma patients, or transcranial magnetic stimulation. And they say that actually, if you stimulate the Brodmann area, you can have an impairment or some in the phonological processing according to Brodmann area 44, and mostly semantic processing in Brodmann area 45. But it seems that there is no evidence for long-term speech impairment, and also as they evaluated, so the the speech, like I can say how for example, in this task, when they are asked to describe a picture, so the spontaneous speech, yes, I wanted to say that. And so this is an hypothesis that like the white matter is like something that maybe is more difficult to recover after stroke. You can, this is so the idea that you can have a more cortical plasticity and for recovering after stroke. So if you have a damage selected selectively to the Broca areas, you can recover thanks to the involvement of the surrounding areas. And so from this data, there is no evidence that can bring to a long-term speech impairment. Instead, if you have a damage on the arcuate fasciculus, and maybe it was also the case of the seminal Broca's patients, you can have, it seems that there is an evidence that you will have long-lasting problems in speech production. And so I found this article really interesting because, of course, they are proposing like a shifting in the view of let's say lesion loading in stroke patients, and of course they focused on the white matter that seems to be really interesting, and maybe if we have a damage of that specific tracts could really impact in long-term patients' deficits. Maybe you have some comments on these or some questions.
Thank you, Leah. We probably got time for one common question. Anyone? Oh, Michelle, yes.
Couldn't help but see that like the lesions that they explore were mostly focused on lesions in the frontal lobe.
Mm-hmm.
And so in order to demonstrate the disconnection hypothesis of the anterior segment of the arcuate fasciculus, you know, you should have a lesion in the parietal lobe that disconnects the same tract and lead to the same impairment or comparable impairment to lesion in the frontal lobe of that tract. And here, I don't think it is the case.
No, they just like drop out patients with lesion on the parietal lobe because they just wanted to focus on the most anterior part of the arcuate fasciculus. So they selected the anterior segment, and they also say that it could be like interesting also to evaluate also which part, like so locally, which part of the arcuate fasciculus could be more related to long-term deficits.
Okay, and I see. And they didn't regress for lesion size.
They consider at some point also the lesion size, yes.
Whilst Leah's looking, this is part of the Plurus study, so they would definitely have the means and the data to look at what you suggested, the disconnection from the parietal as well. So that would be possible follow-up.
Yes, they consider.
Okay, well, that's great.
Okay. But also, it'd be nice to look at all the three segments.
I know.
Right.
The disconnector would, be probably also more data-driven, but yeah, it's great. It's great. Like,
I'm happy to carry on that discussion in a minute, but I don't want to keep people longer than they committed to. So I'm going to end Neuroccino officially here. But you're welcome to hang around and we talk about this paper a little bit more. And I also wanted to draw your attention that starting this week on Thursday, the CNS talks are beginning. So we will have one talk per month on the last Thursday. It's usually at 4 o'clock, but because our first speaker is in Australia, we're going to have it at 10 a.m. this Thursday. And we're going to have James McShine talk about the thalamus. So I hope we see you there. It's the first of many exciting talks. They're all on the website, so do have a look. And they're already scheduled on YouTube as well, so you can set yourself a reminder so you don't miss them. And with that, thank you very much for coming today. Thanks for the papers. And as I said, if you want to hang back a little bit and finish the discussion on this brain paper, then you're more than welcome. Thank you.